Hello friends, so here I will be going ahead with class 12, sexual reproduction of plants, double fertilization. So when we have seen the ovary, when we learnt how the ovary looked like, we saw that this is the micropyle end which has got a opening. So here there are 3 cells. Okay, so we have 2 synergid cells and 1 egg cell is there. Then we have in the chalazal end we have 3 cells, which is the antipodal cells, and in the middle we have 2 nucleus, that is 2 polar nucleus. Okay, so this is the structure of the egg cell. Okay, so what happens? The male gamete or the pollen, it will come and it will fuse with the nucleus. So it will fuse with the nucleus. It will fuse with the nucleus of the synergid cell. So okay, fine. So it will come, it will fuse with the synergid cell. So when it fuses, this is known as syngamy. This is known as syngamy. Okay, so once the syngamy happens, there is a formation of diploid cell, which is the zygote. So there is the formation of diploid cell, which is the zygote. This zygote. Okay, so this the other male gamete, what will happen? This other male gamete will go and will this will go and it will go ahead and fuse with the polar nuclei. So once it fuses with the polar nuclei, as it is 3N, it is known as triple fusion. It is known as triple fusion. Okay, so triple fusion is occurring. So there is 2 fertilization that is taking place. One is syngamy and one is triple fusion. So that is why, as 2 fertilization is taking place in plants, it is known as double fertilization. So what happens here is, so after triple fusion, so since 2 types of fusion, syngamy and triple fusion occurs, it is known as double fertilization, as I told you. Now the central cell after triple fusion, it will become the primary endosperm cell. So this will become the primary endosperm cell. Okay, so we have this 3N, it came and fused here. So this is a triple fusion, and the one that came and fused here, that so it did not, that is not the micropyle end, as usually the micropyle ends are is drawn lower side. So here whatever, because it is 3N, that is primary endosperm nucleus, this is known as triple fusion, and in the micropyle end, as it will come and join the synergid and form the zygote, this is known as syngamy. Okay, this is known as syngamy. Now on this complete fertilization, now there is from this pen or primary endosperm nucleus, a triple fusion, when it occurs, then we will have the primary endosperm cell. We will have the primary endosperm cell, which will develop into the endosperm, while the zygote will get developed into the embryo, like globular embryo, heart-shaped embryo, it will get developed. Okay, so that is occurring. Now coming to post-fertilization event. So post-fertilization, what happens? So in the post-fertilization, after the double fertilization, what happens? There is a maturation of ovule into seeds and ovary becomes the fruits. Okay, so coming to the endosperm, in the triploid tissue, what happens is the cell of the tissues are filled with reserved food. So always the endosperm will be formed first, and then only there will be the zygote formation, because this particular endosperm, okay, that is going to have your food reserve. So whenever there is a growth, then it requires food, it requires nutrition. That is why the food reserve, the endosperm, needs to be there. So in most common type of endosperm development, what happens? The pen undergoes successive nuclear divisions to give rise to free nuclei. And this stage is known as free nuclear endosperm. This stage is known as free nuclear endosperm. Now, after this, there will be subsequent cell wall formation which will occur, and the number of free nuclei formed before cellularization, it will vary greatly. Now what happens if you see in a coconut? So in a coconut, whatever the water you see inside, so that particular is your free endosperm. Okay, that particular is your free nuclear endosperm, and the white shell that you see outside, that is nothing but your endosperm, cellular endosperm. Okay, so that is there. In few cases like maize, pea, groundnut, beans and all those, the endosperm may be completely consumed by the developing embryo before the seed maturation, or sometimes it may exist like castor or coconut. Coconut, we saw how the free endosperm, it exists, the endosperm is existing. Okay, so now now if you split open any fruit or the beans and all those, you can look at the endosperm. Fine. Next, coming to the embryo. What happens to the embryo? So embryo, it develops at the micropylar end of the embryo sac where the zygote is situated. So at the micropylar end, there will be the development of the embryo. Okay. Alright. Now, when this occurs, so there is an adaptation which will provide nutrition to the developing embryo. Though the seed greatly differs, the early stages of embryo development is almost same whether it is monocotyledons or dicotyledons. Okay, so embryogeny is not going to differ. So zygote will give rise to proembryo, then globular, heart-shaped and mature embryo. That all the stages are going to be same. Now in dicotyledons, what happens? In dicotyledons, it consists of embryonal axis and two cotyledons. That is why the dicotyledons, if you have a bean or if you take a groundnut, you will find that there are two cotyledons and there is the embryonal axis that is there. Okay, so the portion of the embryonal axis is above the level of the cotyledons, and this is known as epicotyle area. So this is known as epicotyle. It will have a plumule. It will have a plumule and it will have a radicle. So it will have two things, plumule and a radicle. So plumule is nothing but the stem tip, and the cylindrical portion which is hypocotyle, this is known as the root tip. It terminates in the root tip. So there is plumule, and plumule is the shoot tip and radicle is the root tip. So epicotyle, which is upper area, which is the plumule, and hypocotyle, lower area, which is known as the root tip. Okay, now what happens in the monocotyledons? In the monocotyledons, what happens? So herein they have got the cotyledons. Here is called scutellum. It is called a scutellum. Okay, and that is situated towards one side. So it is situated towards one side, okay, of the embryonal axis. At the lower end, what happens? The embryonal axis has radicle and the root cap, which is enclosed in an undifferentiated sheath, which is coloriza. Now the portion of the embryonal axis above the level of the attachment of scutellum is epicotyle. So epicotyle has got the shoot apex and few leaf primordia enclosed in the hollow foliar structure, which is known as coleoptile. Now coming to the seed. What happens in the seed? So in angiosperms, the seed is the final product of sexual reproduction. So in the angiosperm. So herein the seeds are formed inside the fruit. A seed typically consists of seed coat, cotyledons and embryo axis. So seed coat is there is the cotyledons and the embryo axis. We said that embryo axis have pumule and the pumule and the radicle. Okay, so all these things, and cotyledons, they are a good source of food, and that is why they are thick, swollen. So mature seeds may be non-albuminous or ex-albuminous. So what is non-albuminous? Where there is no residue of endosperm. It is completely utilized by the embryo, like pea, groundnut. So these are non-albuminous. And in albuminous, it retains a part of the endosperm. Okay, it retains a part of the endosperm. It is not completely used up. Like in wheat, maize, it is not completely used up. So occasionally in some seeds such as black pepper, beet, remnants of nucleus is also present. So this is known as perisperm. Perisperm is what? The remnants of nucleus is the perisperm. Non-albuminous, wherein it is completely used up, and albuminous where there is some residual endosperm is there. Okay, next, integuments of ovules are hardened as tough protective seed coat. The micropyle is a small hole that you see in the seeds. You will find a small hole which is the micropyle. So this facilitates entry of oxygen, water during germination. So usually the seed takes some time for it before germination. Okay, so this is known as dormancy. This period is known as dormancy. And once the seed gets the right amount of moisture, oxygen, it will germinate. And when the ovules mature, what happens? When the ovules mature, they will mature into seeds, whereas the ovaries will develop into the fruits. Okay, so the wall of the ovary develops into the wall of the fruit, which is known as pericarp. The fruits may be fleshy as guava, orange, mango, or it may be dry like groundnut or mustard. So there are many fruits which has evolved by the mechanism of seed dispersal. Now, there are two kinds of fruits that might be formed. One is false fruit and one is true fruit. If the thalamus is forming the fruit, then it is the false fruit, and the fruits comes from ovary, then it is known as the true fruit. Okay, so although in most species fruits are a result of fertilization, in few it is not a result of fertilization, and that is when we say the fruit as parthenocarpic fruits. Okay, we say it as parthenocarpic fruits. So banana is a good example of the same. Now, seeds, they offer several advantages for the angiosperm. First is, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. So there is more it is depending more on seed. Okay. Also, seeds have better adaptive strategies for dispersal to new habitats and helps in species colonization. Okay, so next is, there is sufficient amount of food reserve that is there. They can be stored for a very long period of time. Some can be stored even thousands or 10,000s of years. Okay, so then when it gets the right amount of oxygen, water, moisture, it will again go. So there is a hard coat or hard seed coat which protects the embryo. Okay, then seed is the basis of our agriculture. So whatever is the agriculture practice which goes on, they sow seeds and they keep the seeds for the next season. So it is very easy to store seeds. Now coming to what are apomixis and polyembryony. So what is apomixis? Apomixis is seeds, usually we see that it is a product of fertilization. So sometimes in some species like Asteraceae and grasses, they have evolved a special mechanism to produce seeds without fertilization. So production of seeds without fertilization is apomixis. Production of seeds without fertilization is apomixis. Production of fruits without fertilization is parthenocarpy. Okay, so do not confuse between both. So apomixis means it is asexual reproduction, which mimics sexual reproduction. Okay, it's similar to sexual reproduction, but it is asexual reproduction. So there are several ways of development of apomictic seeds. In some species, diploid egg is formed without reduction division. So these develops into embryo without fertilization. So more often, as in many citrus, mango varieties, some of the nucellar cells surrounds the embryo sac, which starts dividing, and they will protrude the embryo sac, and this will develop into embryos. In such species, each ovule contains many embryos. So there is an occurrence of more than one embryo, which is known as polyembryony. So there is more than one embryo, it is known as polyembryony. Now the hybrid varieties of several of our foods, vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased. But there is one problem with the hybrids. So hybrid seeds, they have to be produced every year. If the seeds is collected from the hybrids and sown, the plants in the progeny will segregate and will not contain the hybrid characteristics. So they need to be produced each and every year. It is very much expensive for the farmers to utilize. If these hybrids are made into apomix, there is no segregation of characteristics in the hybrid progeny. Then the farmers can keep on using the hybrid seeds to raise new crops. Okay, so that is one of the things. So that ends the chapter. Thank you.